Yes, good day, class. I welcome you to our revision segment of paper 1, grade 11. I'm sure that we are already preparing for our final exams of the year. So this is a full paper for paper 1. Let's go through it. I don't want to waste any time. Just check through. We are going to do the full paper. So just be patient. It's going to be long, but just follow it through to the end. So therefore you can be able to get everything right and correct. We're just gonna go straight to the questions. Uh, First question, section A, we are given various options that we are to choose, and then choose the correct answer and write only the letter next to the question number. Please follow the instructions per question. Do not make any mistake. We are given an example, in this case 1.1.11, which is given like to say you just write the question number and then the letter that is correct for that answer, for that question. Please make sure that you don't make mistakes as you go per question. So first question, cellular respiration in green plants takes place. Cellular respiration in green plants, in green leaf takes place A, continuously, B, during the day only, in tissues without chlorophyll only, D, during the night only. So I'm saying if you can look in this question, get the correct answer, the best suitable answer in this is what is cellular respiration in a green leaf takes place continuously. It takes place continuously. So our answer is what? Is A. Our first question, our answer is A. It takes place continuously. Please don't make mistakes. Our answer is what? Is A. It takes place continuously. And air breathed out is different from air breathed in because it is cooler. A. B, it is drier. C, it contains less carbon dioxide, contains less oxygen. Air breathed out is different from the air breathed in because it contains less oxygen. Air that is breathed out contains less oxygen. So our answer is what? Is D. It contains less oxygen. Please make sure that you don't make mistakes. I'm saying follow the answers correctly. It is D. Air breathed out is different from the air breathed in because it contains less oxygen. Next question, which one of the following can be absorbed without further digestion? Which one of the following can be absorbed without further digestion? A, can be absorbed without further digestion. A, proteins, no. B, lipids, no. C, glucose, yes. And then obviously starch cannot. So our answer is what? Is glucose, which is what? Which is C. Which is C, can be absorbed without further digestion. Glucose can be absorbed without further digestion. 1.1.4, photolysis occurs during 1.1.4. It occurs during A, dark phase, B, light phase, C, Krebs cycle, and D, oxidative photolation. So what is happening is that when does this occur? 1.1.4, it occurs during the light phase. It occurs during the light phase, which is B. Our answer is B. It occurs during the light phase. Let's just go to the next question. Which one of the following characteristics of a developing country is false? Which one of the following characteristics of a developing country is what? Is false? Which one of the following characteristics is false of a developing country? Number 5, high birth rate, a developing country. Which one is false? Please take note, the question is saying is what is false. So we are saying is false is where you should look at. It has got high birth rate. That is very correct. A developing country has got high birth rate. B is high infant mortality rate, it is not false, it is correct. C, high standards of living, that's not correct. High population growth, so which is false is that a developing country does not have high standards of living. It has got low standards of living, hence the answer is what? Is C. And then the next question is, it is a question that is to 1.1.6 to 1.1.7 are based on a diagram showing showing the human thyrox. And then the question wants us to do what? It is human thyroxine. Which of the following are represented by A, B, C, D? Which of the following are represented by A? Are represented by A. We are saying what is represented by A? A. The question wants us A, B, D, and E. So what is represented by A, D, and E? It should be A. We are saying what is represented by A, D, and E, which is number 6. As you can look, A, B, D, and E. I always tell my class that when you look at questions such as this one, you must be able to see that where you don't get the answer, where some of the answers you don't know or letters you don't know, at least mark to say that you know at least 2 or so, which you should use to select the best suitable answer. So what is happening is that what is represented by A, B, and E, A, B, D, and E. So we are saying it should be A should be the larynx. A should be the larynx. And then B is what? Is the trachea. A it is the A. And then B is the what? B is the trachea. And then what is it? Which is the D is actually showing something inside the cavity there, or it is the lungs. And then what is AB is saying A, B, D, and E. And then E should be, where is E? Our E. Okay, E obviously should be intercostal muscles. It should be intercostal muscles. Intercostal muscles. It should be a intercostal muscle. So from our points there, we are saying what is represented by A, B, and E. So our A, we said, is the larynx. Our B is the trachea. Our D is the lungs. And then our last one is the intercostal muscle, which is correct as to say that the answer is what? The answer is B. And then is B. Our answer is B in this regard. It is B. Next question. Study the features listed below. Number 7. Lined with A to remove dust. 2 is cartilage that is cartilage that are C-shaped to allow the esophagus to stretch during swallowing. And then 3 is lined with ciliated columnar epithelium tissue with globular cells, secret mass mucus to trap dust and germ. Which combination of features are adapted to part B? B of which we said is the what? Which we said our B. We said our B sorry. We said our which combination is attracted to B, which is the trachea. What is the one that is a combination of letters that are explaining the trachea in this regard? So which one is explaining the trachea in this question? Number 7. We are saying it. Which one of these letters or which one of these characteristics explains the what abducted to B, which we said is the what is the trachea? Which one is explaining the trachea in this case? So if you look in this, we are saying it should be the trachea should be it's got a cartilage. It's got a cartilage that are C-shaped. It has got a cartilage that is C-shaped to allow the esophagus to stretch during swallowing. That is very correct. A trachea has got number 2 is correct, but it has no there is no way to remove dust in what in this, which is wrong. One does not exist. Lined with ciliated column epithelium cells, tissue that goblet cells secrete mucus to trap dust and germs. And then we are saying it is also correct. So our answer is what is C, which is 2 and 3 only. 2 and 3 only. They are the ones which explains the trachea. It has got the cartilage C-shaped, and also it has got the globular cells that secrete mucus to trap dust and what and germs. That is correct. Number 8, which of the following is the correct sequence of activities that occur during nutrition? Which of the following is the correct sequence of activities that occur during nutrition? So in this case, we are saying what should happen. Firstly, the food must be eaten. That is, ingestion must take place. So in this case, we are saying ingestion must be the first stage. So here we are saying from all these options that are given, we cannot have absorption, we cannot have digestion, we cannot have release before eating. So ingestion must be correct. So we are saying our answer should be B, which should have ingestion, digestion takes place, and then absorption takes place. Lastly, we have got ingestion, which is the release at last. The residue is lost, is released out. So our answer is what is B, is the correct sequence of activities that occur during nutrition. Each of the following increase the rate of photosynthesis except an increase in dash. Which one does not have which of the following each of the following increase the rate of photosynthesis except increase in dash? Number 9. Which one does not we are saying can increase photosynthesis except increase in what? Photosynthesis, carbon dioxide is needed for photosynthesis, light is necessary for photosynthesis, and also even which we are saying oxygen is produced during photosynthesis. So it is not increasing, it is produced. And then we are saying what is the temperature? Obviously, sun intensity, light, which is there. So our answer is what is C. C is the answer, which is does not increase, or does an increase in oxygen does not increase the rate of photosynthesis, but oxygen is a byproduct actually produced when photosynthesis has occurred. So our answer is C, number 9. Next question. Give the correct biological term for each of the following descriptions. Write only the term next to the question number in the on your answer book. Write only the biological terms. Class, understand. The question wants you to give biological terms. It does not need you to write statements. It's scientific explanation or scientific definition for these statements that are given. So storage form of glucose in plants, what is it called? It is called what? It is called starch. It is called starch. The lymph vessel which transport absorbed fats, what is it called? The lymph vessel which transport absorbed fats. We are saying it must, you know very well, it is called what? The lacteal. Wave-like muscular contractions which move food along the alimental canal. The contraction and relaxation of this, the digestive system or the alimental canal is called what? It is called peristalsis. It is called peristalsis. It is called peristalsis. A nutritional disease which occur in children who are fed on a high carbohydrate and low protein diet. When children have got more of are fed of food which has got more carbohydrates or high carbohydrates and low proteins, it results in a condition called what? Kwashiorkor. It results in kwashiorkor, where you see the child would have a big head, small neck, big stomach, and also is mass wasted, very skinny, pale, and stuff. So we are saying it is called kwashiorkor. A protective membrane surrounding the lungs. What is it called? It's called the pleura. A structural and functional unit of the kidney. The structural and functional unit of the kidney. I'm sure we have talked about this also in our various lessons, which is called the what? The nephron. Is the structural and functional unit of the kidney. And then energy carrier in cells, it is called what? ATP. I'm sure we are still moving together. Please don't get lost and follow up as close as possible. The acid which accumulates in the muscles of a human during continuous strenuous physical activity is called what? It's called lactic acid. It's an acid that accumulates in muscles of humans during continuous strenuous activities. So it is called what? It's called lactic acid. It is called lactic acid. That is called lactic acid. So please, these questions, I always encourage class that you should be able to get the best marks out of this. Get as many marks as you can. Actually get total in definitions. Indicate whether each of the following statements it applies in column A applies to A only, B only, both, or none. So in this case, you must write your answers very correctly and make sure that again, for example, we are saying 1.1, 1.3.1. You are supposed to write the question number and the answer only. Write only the answer. So microorganisms which cause tuberculosis, what is it called? Tuberculosis is caused by what? By a bacteria only. It is a bacteria. So our 1.1, our 1.3.1, it is a only. So I'm saying, please take note. I'm saying it is a bacteria. So this is, I'm just illustrating on the first here, on the first question here, so you can see what how you must write your answers. It is a only. Okay, in the mitochondria. What occurs in the mitochondria? We are saying the Krebs cycle, it occurs in the mitochondria, and also the oxidative process also it occurs in the mitochondria. So it is what? It is both A and B. Both A and B is correct, is correct, is correct. It's both A and B in number 1.3.1. And then organ which secretes gastric juice. Which organ secretes just gastric juice? Organ which secretes gastric juice. Gastric juice is not secreted from the liver. It is not secreted from uh it is not secreted from what? From the pancreas. So in 1.3.3, it is what? It is none of these. It is none of these. Gastric juice is not secreted from the what? From the liver. It's not secreted from the pancreas. So please take note that you don't miss you don't write and you don't get the answers wrong. Density independent factor number 1.3.4, density independent factor 1, uh, drought is density independent, also floods. So number 1.3.4 is both A and B, which we are saying both A and B is correct. Individuals of the same species competing for resources are what we call that. Individuals of the same species competing for resources. What is it called? It is called what? Intra competition. Inter, it means different species. Intra is within the same population. So it is what? Intra competition, which is B only. It is B only. It is B only. So I'm saying intra is within the same species, and then inter is across species. So please, these are also some other questions that you should not miss. And then I told you again to say when we say inter, it means outside specific uh, limitations that are within the group. And then intra is within the same group. Intra competition is grade 11 life science students alone competing amongst themselves. It is intra competition. Who is getting number 1, number 2, number 3? And then when we are going to say as a district, it's inter competition, where we are competing schools, school A versus school B. It is inter competition there. The diagram below represents it represents a age population distribution pyramid for human population of a developing country in 1 year at a certain time. This is a population pyramid that is shown in this question, population pyramid. So we have got our age group 0 to 85 plus, and then we have got a percentage of females there, and then we have got percentages of what? Of males on the other side, and then pyramid A. So let's just quickly check the question. Comment on why the above diagram comment why the above diagram represent an age pyramid of a developing country. I'm sure we have had a question similar to this one in the previous section there. Why is the above represent an age pyramid of a developing country? One, it has got high birth rate, which we are saying it has got what? A broad base. It has got question 1.1.4, 1.4.1, 1, the pyramid 1. In this case, what is happening is that our pyramid is of a developing country because it has got a broad base that I'm sure that we can as a class agree to that, that there is a what? There is a broad base there. We have a broad base that is shown there. The pyramid is a very broad base, as you can see from there to there. We are saying the base, the pyramid is a is a it has a very broad base. It has a very broad base, due to high birth rate that we can talk about. Broad base due to high birth rate. Due to high birth rate, the high birth rates in the older age group. Also in the older age groups, fewer individuals survive, as you can see. In older, number 2, in older age group, fewer individuals are surviving. Older age groups, fewer individuals are surviving. As that you can see, in the older age groups, fewer individuals are surviving. If you can see, in a developing country, there is a problem of life expectancy or low life expectancy, where we can see that as we can see, from the age of 45, from 45 year going up to 85, you can see our numbers are decreasing. There is a decreasing survival rate here. This is due to factors that are quite a lot of them. It can be because of poor hygiene, poor health facilities, heavy work stuff that the people are doing, which can also cause them to get old and suffer and die early. Is talking about poor mechanization, which is making people to work in industries and workplaces that are difficult and so forth. Number 1.4.1, name the method used to determine the population size of a country. Name the method that can be used. Name the methods used to determine the population size of a country. I'm sure that as a class, we understand that census is done whereby the number, the country's population is counted. People are counted to see how many people do we have in a country. So population is determined through census. 1.4.3, why is the life expectancy lower in a developing country than a developed country? Why do we have to have lower life expectancy in a developing country than a developed country? It can be one, low life expectancy in developing country is caused by various factors. Amongst them, we can say one, it can be due to lack of medical care, lack of proper medical care, which can say the medical care cannot be enough, that people sometimes they end up dying young. And also it can also be due to low standards of living. Low standards of living. Low standards of living make people to die early. And then it can also be due to the standards of living. This we can talk about quite a lot of examples, e.g., we can talk about sanitation. We can talk about sanitation. We can talk about sanitation. We can talk about the hygienic conditions. These are standards of living. Even number 3, we can also talk about poor nutrition. Poor nutrition. Even poor nutrition can cause people to die early also, because when people, they don't eat balanced diet, they don't eat, if people, they don't eat nutritious food, they would also end up dying early and so forth. So these are some of the reasons which can cause which can cause low life expectancy in developing country. And even again, I've given another example, such as uh, poor mechanization. in what? In industries. Results in people doing too much heavy work. Results in people working bad conditions. People working under bad conditions. Which of these conditions might cause them to die early as well? So what we are saying is that these conditions, they might as well affect the living standards or life expectancy of what? Of our people. People can end up dying early because of such a condition that we are referring to here. So what is happening is that we need to understand the characteristics of developing and developed country. The following diagram illustrates some investigations carried out on photosynthesis. The plant was kept in dark for 48 hours before being placed in sunlight. The plant was left in the dark for 48 hours before being placed in what? In sunlight. And then our first question is saying, these are apparatus that are given the leaf C, leaf B, aluminum foil, and then we've got leaf A, potassium hydroxide solution, and then apparatus to investigate aspects of photosynthesis. First question is saying, what is the aim of the investigation on leaf A? Leaf A, what is we are saying this plant is there, and then it is within a jar which is covered. There is a jar that is covered there in leaf A. Our leaf A, it is inside the jar. So what we are saying is leaf A, it is inside the jar. So what is the purpose of this investigation there? What's the aim of the investigation on leaf A? Leaf A, therefore, we can see that it is there to investigate whether light is required for photosynthesis or not. A, to determine if light is necessary for photosynthesis. To determine if light is necessary for photosynthesis. That is the reason of leaf of the plant to have the leaf A inside the jar there, to determine the necessity of light for photosynthesis. And then leaf B, what is happening there? It is aluminum foil. Part of it is covered with aluminum foil. So what is happening? We want to investigate whether carbon dioxide is what is required for photosynthesis again. To investigate leaf B, to determine if carbon dioxide is necessary for photosynthesis. It is to determine the necessity of carbon dioxide for photosynthesis class, leaf B. You see that aluminum foil that is there. So it is actually obstructing the absorption of carbon dioxide by the leaf there. Hence, we would need to identify if carbon dioxide is necessary for photosynthesis. Number 1.5.2, why was the plant kept in the dark for 48 hours before being placed in sunlight? Why was the plant left in the dark for 48 hours before? Why was the plant kept in dark for 48 hours before being placed in sunlight? Basically, darkness is done in order to discharge the leaf, to discharge the plant, to discharge to discharge the plant. It was done to discharge the plant, to remove starch from the plant. That is the reason why it was put in the what? In the dark for 48 hours. And then what is the purpose of the potassium hydroxide solution? Potassium hydroxide solution, what is it for? It is actually used to absorb carbon dioxide from within the bell jar there. The potassium hydroxide, as you can see there, the potassium hydroxide, which is here, what is its function? 1.5.3, 1.5.3, it is to absorb carbon dioxide from within the what? The bell jar. To absorb the carbon dioxide from the bell jar. That's the purpose there of potassium hydroxide. And then number 1.5.4, the following questions refer to the method used to test the leaf A, B, and C for starch. Why number 1.1, 1.5.4.1, the next this question, why was the leaf boiled in beaker of water? Why was it boiled in beaker of water? Actually, boiling is done to soften the leaf, or to soften the tissues of the leaf, or to break down the cells, the cell walls, to soften up the tissues, to soften up the tissues, or we can say to break down the cell walls of the what? Of the leaves, to break down the cell wall of the leaf. That is why it was boiled. That is why boiling is done to soften the tissues of the leaf, or to break down the cell. Again, the next question is saying, why was the leaf boiled in test tube containing alcohol? Alcohol, it is done to extract to remove chlorophyll. Alcohol is done. Boiling in alcohol was done to remove chlorophyll. To remove what? Chlorophyll. That is the reason why it was what? That's the reason why the plant was boiled was boiled in a test tube containing alcohol. Name the chemical used to test for starch. What is used for starch is tested using what? Using iodine solution. It is iodine solution that is used in this case. It's iodine that is used in testing for what? For starch. Uh, Please just take note that questions of photosynthesis and so forth is quite common and is quite used in our various questions. So if you are preparing for your national any exam, make sure that you cannot go to exam without understanding these experiments. For photosynthesis is carbon dioxide necessary for photosynthesis, is chlorophyll necessary, and also the other experimental conditions, such as to say some conditions are set up without manipulation, which we are saying unmanipulated settings are used as what? As the control of the investigation. They are used as measurement, as standards of reference or measurement. You would find questions such as those any other time. So please take note. The table below shows the effect of light intensity on the rate of photosynthesis in a plant when placed in two concentrations of carbon dioxide. We have got light intensity, rate of photosynthesis in 0 carbon dioxide, in 0.03 carbon dioxide, and also 0.13 percentage carbon dioxide. So we are given this. Here it's 0.9, 1.2, 1.5, 2.0, 2.3, 2.4, and then it became constant from 5 up to what? 5 up to 7, as well as the other one also became constant from the same level, from what? From 5 up to what? From 5 up to 7 was also constant. So let's just quickly go to the question just to save time. Using the results reflected in the table above, draw on the same system. Other questions can say same system, others can say same set of axes. Ne? So please don't miss the question. Same set of axes or same system of axes, line graphs. The question wants us to draw line graphs showing the rate of photosynthesis in the two consecutive concentrations of carbon dioxide when exposed to different light intensity. So you are drawing two graphs here. We are supposed to draw two graphs which are we are supposed to draw two graphs here. So in this case, obviously the graph is there. These are line graphs which are supposed to be showing the concentration, the rate of photosynthesis at 0.03 and also 0.13%. So in any case, you must understand that a graph must always have what? Must always have a title. So I'm just going to do it here for the interest of space, and let me just draw it here. We are saying the title, our title is showing, is going to be the line graphs. It's supposed to be line graphs showing these are line graphs showing the rate of photosynthesis, showing rate of photosynthesis we are looking at rate of photosynthesis in 0.3%, in 0.03% carbon dioxide, 0.03%, and 0.13% carbon dioxide at different light intensity, carbon dioxide at different light intensity, at different light intensity. So in any case, when you are drawing a graph, there should always be a caption. The title must be there. Line graphs. There are two line graphs. So I'm saying line graphs showing rate of photosynthesis in 0.0%, 0.03%, and 0.13% carbon dioxide. Carbon dioxide at different light intensity. Therefore, that is it. And then we are supposed to understand that our graph must be what? Our graph must, we need to draw our graphs here. So what is happening is that the graph must not be too small. It must not be too big also. So here I'm just doing it for the interest of illustration and just for us to understand what we are saying. Therefore, what must be on the vertical axis and what must be on the horizontal axis? Before there, we can look at the interval of our figures. We have got from 0.9 is the least up to what? The highest we are saying is the smallest. This one is the smallest set of values, and then is the what? Is the highest. So what I'm saying is that when we are drawing the graph now, we must make sure that our graph must have what? Must have must accommodate this set of values there. It must accommodate the set of values that we are looking at. So here now, I'm saying there must be a scale. There must be a scale that can, so therefore the graph cannot be what? Cannot be too small, cannot be too big. So I'm going to draw it like this. I can just say, so on the horizontal is light intensity. Light intensity is given there. And then on the vertical axis, we have got what? On the vertical axis, we should have rate of photosynthesis here. We should have rate of photosynthesis. Rate of photosynthesis on the vertical axis. So I can just choose a scale of saying this is 0. And then we can say 0, 1, 2, 3. We can say 0, 0. Let me see. 0, 1, 2, 3, 4, and then 5 there. So I'm saying I just choose a specific scale of representation of my set of data as to say 1, 2, 3, 4. In terms of this, this graph here, 1, 2, 3, 4. I'm just saying. So therefore, we have got light intensity, which is ranging from 1 to 7. So I can start with the first one, 0.03. 0.03. I can start with 0.03, which is we are saying from light intensity of 1. What we have at 0.03, it was 0.9. 0.9, I can say 0.9 is somewhere there. I can say 0.9, and then at 2, it's 1.5. So 1 here. So 2, it is what? 2, it is 1.5. 2 is 1.5, so my 1.5 can be there. And then 3, it is what? 2.0. 3 is 2.0, which is 2.0 is somewhere there. And then we've got 4. 4 is how much? 4 is 2.3. 4 is 2.3, which is somewhere a little bit up there. And then I've got 4. 5 is 2.4. 5 is 2.4. Also a bit up there. And then I have got 4, 5. 6 is 2.4. 6 is 2.4. And then 7 again is what? 7 is 2.4. So I'm just drawing my graph like this, joining my points there. So this is my graph here. I can just leave it like that. I will show you now to say that the difference, the other one, I'm going to make it broken. The other one, I can make it solid. And then this is the one for 0.3. and then 0,13, it is 1,5. It is starting at 1,5. It is starts at 1 is 1,5. Which is starting there. 1,5. And then I'm saying the next one is what is 2,3. 2,3, which is somewhere there. 2,3, and then it is 3.1. 3.1 is somewhere there. And then it is 3.1, it is 3.9 at 4. 3.9 at 4, somewhere there. And then at 5, it is at 5, 4.3. 4.3, 4.3, and 4.3. So I also have my graph, which is starting and then going. So I'm saying this, I can also just make it to say 0.13%, and then this is 0,03%, 0,03%. So as you can see there, this graph can be, it's just on the same set of axes. It's just on the same set of axes that you can see. 0,13 and 0,03%. They are all drawn on the same set of axes there. They are all drawn on the same set of axes. In any other times, you might find out that sometimes we can as well put our readings there to say this is 4.3, this is 4.3, this is 4.3, and then it is 3.9, 3.9, 3.1, 3.1, and then 2.3, something like that. So what I'm saying is, in other cases, you need just to mark the points that we have put the readings of the point that you have drawn, just to ensure that the person marking can be able to see what you are saying, instead of trying to assume to say what is the point marked there representing. The same applies here to say that we can as well put the readings to say this is 2,4, this is 2,4, this is 2,4, so that we can see. I'm saying each of the markings that are on the graph there can be given their vertical readings, and so therefore it can be easy for the marker to see what is the meaning of what you are saying. So these are the line graphs that we have drawn there. These are the line graphs that we have drawn. As you can see there, they are on the same set of axes. Vertical axis is rate of photosynthesis, and then horizontal axis is light intensity, which is also labeled and showing, as you can see on our graph there. So second question is saying, describe two conclusions that can be made from the graphs. What conclusions can be made from the graphs here? We can say 2.13, 2.1.2. Two conclusions that can be drawn there. The rate of photosynthesis is higher at carbon dioxide concentration of 0,13%, as you can see. I'm sure that you can see the graph that is on top there. Rate of photosynthesis is higher on the graph that is 0,13%. I'm sure that you can see on my graph there, which is shown by what? Which is shown, this is this is our graph that we are talking about. 0,13% rate of of photosynthesis is higher, rate of photosynthesis is higher, at carbon dioxide is higher, at carbon dioxide concentration of 0.13%. At a percentage of 0.13%, which we can see class there, 0.13% than 0.3%. That is quite correct there. And then we can also say for both carbon dioxide concentration, the rate of photosynthesis increases up to light intensity of 5, and then it became constant both. As you can see on here, from the graph there, I said this, it became constant, and also it came also constant also on what? On this one, which is like that. So the other conclusion that can be said that is that that can be said is that for both carbon dioxide concentration, for both carbon dioxide concentration for both carbon dioxide concentration concentrations, what happens? The rate of photosynthesis, the rate of photosynthesis, the rate of photosynthesis, the rate of photosynthesis happened. What happened with it? Increases up to what? Up to the light intensity of 5, and thereafter it became constant. As you can see there, rate of photosynthesis increases. It increased up to light intensity, up to light intensity uh, of 5, up to light intensity of 5, and thereafter it became constant. So questions such as this class, what happens there is that you are supposed to conclude not on your thinking, but you are just telling the examiner what you are seeing on the graph. You are just saying what the graph is showing. You don't decide, you don't think. You are giving the representation of the results, what the results are representing. So in both cases, carbon dioxide, at both cases of carbon dioxide concentration, the rate of photosynthesis increased it increased up to light intensity of 5, and thereafter it became what? It became constant. It became constant, that we can see. The next question suggests a reason as to why the rate of photosynthesis would drop if carbon dioxide levels is increased to 4%. How can it, or why should it drop if that it increased up to that level? So what happens here is that 2.1.3 2.1.3 what happens here is that higher concentrations of carbon dioxide, higher concentrations of carbon dioxide in cells, increases the acidity, increases the it increases the acidity and lowers the pH. And lowers the pH, causing enzymes to what? Causing enzymes controlling the dark phase to become what? Denatured. Causing pH, causing the enzymes it causes the enzymes it causes the enzymes that controlling the controlling in the dark phase to be destroyed or killed. To be destroyed or killed. That is what is happening there. So in that regard, the rate of photosynthesis will drop eventually. Rate of photosynthesis will drop. The rate of photosynthesis will therefore drop. That is what happens there. If we can increase the carbon dioxide, if we can increase to 4%. So as you can see, we are saying that is going to be the problem there. That will cause the problem to what? The photosynthesis to drop anyway. So let's quickly just go to the next question. So concentrations of carbon dioxide in the cells will increase acidity and lowers the pH. It lowers the pH here. Potential hydrogen atoms, I'm sure that the acidity and alkalinity of any given condition, which we call the pH, and then causing the enzymes controlling in the dark phase to be destroyed or killed, and then the reaction therefore will decrease, which will eventually reduce the rate of photosynthesis. Our next question. The diagram below shows human digestive system. The diagram below shows human digestive system, and then it wants us to say it is the human digestive system. Give the letter and the name of the part that produces bile 2.2.11. The letter, give the letter. The question wants you to understand, give the letter and the name of the part that produces bile. So which part produces bile? It is the letter and the name. We are saying letter, it is 2.1.1. It is E, and then the liver. It is E, and then the liver contains millions of tiny finger-like projections called villi. What is that? It is number next. contains millions of tiny finger-like projections called villi or ovillis. What is it called? It is in the small intestines, which is D, and then small intestines, and then a big like organ which store food temporarily. Obviously, it is the stomach. Which one is the stomach? A big like organ. It should be what? It should be A. A is the stomach. We are saying it is the stomach that stores food temporarily, which is a big like organ, the stomach, and then state the end products of lipid digestion. It is the end product of lipid digestion. It is fatty acids and glycerol. In this case, it is fatty acids and what? And glycerol. Protein digestion. The end product of protein digestion, it is what? It is amino acids class. When proteins are digested, their end products is what? Amino acids. It's amino acids. Identify organ B. Identify organ B. That one. Organ B is the pancreas. Organ B is the pancreas class. This is the pancreas. Organ B is the pancreas. It is the pancreas. That is organ B. And then explain the role played by B when the blood glucose level is raised above the normal limit. I'm sure we understand the balancing of what? Of glucose in the body on this regard. Next question, this next question, we are saying what is the role of the organ B when blood glucose is raised above the normal limit? When blood glucose level is high, when blood glucose level is high, the organ which we are saying the pancreas secretes insulin. The pancreas or organ B, it secretes insulin. It secretes insulin into the blood. It secretes insulin into the blood. Insulin decreases the blood glucose level. The insulin will decrease the blood glucose level. It will decrease the blood glucose levels by converting that excess glucose into glycogen. By converting excess glucose into glycogen, which we are saying, which also promotes the absorption into glycogen. I'm saying it actually converts converts the glucose into excess glucose into glycogen, which is the storage form of glucose, into glycogen, which can be stored, in the liver. So in that regard, it is stored in the liver and what? And muscles. And then what is next is that it also promotes the what? The absorption of glucose into the what? Into the body cells. It also promotes absorption of glucose into what? Into the body cells. So that is how the pancreas works when what? When the blood glucose is high, it activates, I mean, it is activated to secrete insulin, which will help in converting excess glucose into what? Into what? Into glycogen, and then which can therefore can be stored in the what? In the liver and muscles. The pancreas would also I mean that insulin would also stimulate or promote the absorption of glucose into what? Into the body cells, therefore reducing that high glucose level. So this, I'm sure that we have seen in some books, which can show the summation, uh, which we can show the summation of this process. So here we can say it's insulin, and then here it is what? It is glucagon. So what you should understand is that insulin, it decreases glucose. It decreases glucose levels by converting it into glycogen. And then this one, it increases. It actually increases glucose level. So please take note of this, such that you don't make mistakes. Insulin, it decreases glucose level, and then glucagon, it increases it. So this is a summation of the processes in terms of the balancing of glucose in the body as a result of the insulin and what? And glucagon. So I'm sure that you can understand, and you can as well be able to relate. Let's just quickly move to the next question. A test tube containing some yeast and sugar was placed in a water bath as shown in the diagram below. After 10 minutes, the rate of bubbling was measured. The experiment was then repeated with temperature of the water bath at each temperature shown in the table below. Test tube A has a layer of paraffin to prevent oxygen from reaching the yeast and sugar solution. And then we are given the diagram there. Test tube A and B, bubbles of gas, paraffin there, and then sugar and water bath there. So delivery tube, we are given temperatures and number of bubbles per minute, temperatures and number of bubbles per minute. So we are given there. The question is saying, name the type of aerobic respiration taking place in yeast cells, which produces the gas. name the what? Name the 2.31. Name the type of aerobic respiration taking place in yeast cells, which produces the gas. It is what? What is the type of this? You can you can see we are having yeast and sugar there, and then we have got water bath. So what is happening is that this type is called what? Alcoholic fermentation. This is alcoholic fermentation that is being shown in this respect. It's alcoholic fermentation. And then identify the the dependent and independent variable. Identify the identify the 2.3.1, the dependent and independent variable. So I always refer to my lesson. I tell my students that when you are looking at this question, we would see whether what is being investigated and what is controlling what. The experiment was then repeated. A test tube containing yeast and sugar was placed in water bath in a diagram below. After 10 minutes, the rate of bubbling was measured. The experiment was repeated with the temperatures of the bubbles bath at each temperature shown in the table below there. So what we are saying is that in this question, what is our identify the dependent variable? Our dependent variable is what? Our dependent variable is it is the what? The carbon, it is the carbon dioxide or oxygen produced. The gas produced is dependent. It is the oxygen produced. What is it? The amount of carbon dioxide produced. The amount of carbon dioxide, the amount of carbon dioxide produced here, it is the what? It is our dependent variable. And then, which is also the independent is what? Is temperature. As you can see, at different temperatures, the amount of bubbles were measured. So we are saying it is what? The independent variable, it is the temperature. Temperature is the independent variable, which is question next. So amount of carbon dioxide produced is depending on the temperature available. As you can see on the graph there, we are saying temperature and then the bubbles produced per minute. So the bubbles are what? Are depending on the temperature that is being applied. So we are saying our question, our answer are corrected or are taken from the items that are in question, the temperature, concentration, which is affecting the number of bubbles that are produced per minute. And then name the gas being produced here, the gas that is produced when fermentation takes place. Obviously we are saying it is carbon dioxide that is produced. Carbon dioxide, it's carbon dioxide. It is carbon dioxide. Carbon dioxide is produced in this regard. Describe the test used to identify the gas present in the test tube. Describe the test. Describe the test used to identify the gas. Describe the test used to identify the gas present in the test tube B. So we are saying in regard to this, how do we introduce the gas into a test tube that is containing clear lime water, 2.3.4, introduce the gas. into a test tube containing clear lime water and then if the clear lime water if the clear lime water if it turns milky, if the clear lime water turns milky or become cloudy then the gas tested for is what? Then the gas being tested is carbon dioxide. If we are saying we introduce the gas into a test tube containing clear lime water, then if the clear lime water turns milky or become cloudy, then the gas being tested is what? Is carbon dioxide. The gas being tested is therefore is carbon dioxide. Last question, on the same question, 2.3.5. Explain why the number of gas bubbles released per minute decrease when the temperature is 60 degrees Celsius. This is because high temperatures it is because high temperature it causes the enzymes controlling high temperature causes the enzymes controlling the processing. It causes the enzymes controlling the process of anaerobic respiration. The anaerobic respiration to what? To become what we call denatured. They become denatured and then the reaction decreases. The reaction rate decreases, which resulting in a decrease in the number of bubbles, resulting in a decrease in the number of bubbles. You understand? So what we are saying is that an increase in temperature, why the number of bubbles decrease, the number of bubbles released per minute decrease when temperature is 60 degrees Celsius is because the temperature will destroy or will kill the enzymes that controls the anaerobic respiration, and then the reaction therefore decreases, which would result in a decrease in the number of bubbles released. That is one reason why this is to take place like that. As you can see from the graph, at 60 degrees Celsius, the number of bubbles decreases. It went up from 10 up to 50, but at 60, the number of bubbles decreases. Hence, it means that the anaerobic respiration enzymes are destroyed. The diagram below represents a section through an alveolus surrounding blood capillary in human body. It is showing the alveolus surrounding. It shows the section through an alveolar surrounding blood capillary in human body, which is shown there. I'm sure I once also presented this one. So the question wants us to say, first question, name the type of epithelium tissue numbered 1 and 2. What is the epithelium tissue that is numbered 1 and 2? It is called the what? It is called the squamous epithelium. And then next question is saying identify the blood cell numbered 3. What is the blood cell numbered 3? The blood cell numbered 3, it is called the what? The red blood cell. It is called the red blood cell numbered 3. This is a red blood cell, which we sometimes also call the red blood capsule. Some can say red blood. That is number 3. You understand? And then next question is saying, discuss one structural adaptation displayed by the blood cell identified in, that is the red blood cell, which enables it to carry out its function efficiently. The question is asking to say, how is the red blood cell adapted to perform its function properly? The adaptation of the red blood cell to perform its function. Question 3.13, 3.1.3, adaptation of red blood cell. Adaptation of red blood cell. How is it adapted to perform its function? So one, we can put as many points as we can. One, it doesn't have a nucleus. It does not have a nucleus, making it making it to have more spacing. to have more space, making it to have more space available to accommodate more, making it to have more space available to accommodate more space to accommodate more hemoglobin, to accommodate more hemoglobin, to take up more oxygen. So what we are saying is that the red blood cell does not have a nucleus, which makes it to have more space available to accommodate more hemoglobin, such that it can take more oxygen. And then again, it is very flexible. It is very flexible. As another point, it is very flexible and can twist and bend itself out. Very, very flexible and can twist and bend itself out. And bend itself out of shape to fit into narrow blood diameter, to fit into blood capillaries, which have got with a narrower diameter. With a narrower diameter, that is also one other point, slowing down its movement, allowing for more time to take up oxygen. Slowing down its movement, allowing for more time to take oxygen, allowing for more time to take oxygen. So what we are saying is that the red blood cell is very flexible and can twist and turn, as well as bending to fit itself out of shape, bend itself out of shape to fit into blood capillaries that are even very narrow, with a very narrow diameter. That will be also slowing down its movement, which eventually allows it more time to take oxygen. That is another reason why we can say that it is like that. Another last point that I can also add is that it is biconcave to increase the surface area so that more oxygen can enter. It is biconcave. It is actually biconcave to increase the surface area to increase the surface area so that more oxygen can enter or diffuse into the red blood cells. Surface area so that so that more oxygen can diffuse or enter into red blood cells. So that is the adaptation of the red blood cell to perform its function class. The red blood cell is adapted to perform its function because of these factors that I've listed here. They are actually, I've given you these different points as to say, how is the red blood cell adapted to perform its function? So please take note and don't miss it. Discuss the difference in composition of blood entering at A and leaving at B. Question 3.14. Discuss the difference in composition of blood entering at A and leaving at B. The differences with blood entering at A and also going out at what? Going out at B. So what is the difference? What is the difference in composition of the blood that is entering, the blood that is entering at A and also the blood that is leaving at B? So difference in blood entering at A and leaving at B. What's the difference? The difference in blood entering at A and leaving at B, the blood at A is deoxygenated. Blood at A is deoxygenated, meaning to say it does not have oxygen. It is deoxygenated and or it also has a high concentration of carbon dioxide and a more concentration, of carbon dioxide. It also has a more concentration of carbon dioxide while at B. What happens at B? Well, at B is what? It is oxygenated. At B, the blood is oxygenated. There is oxygenated blood here. It is oxygenated and then it is deoxygenated. It is deoxygenated at A and oxygenated at B. So that it is deoxygenated, it is oxygenated and also has got high concentration of oxygen. High concentration of what? Of oxygen. Oxygenated and then deoxygenated also, we said again, more what? More carbon dioxide in terms of this one. And then oxygenated and then more oxygen than carbon dioxide, more oxygen. So as you can see, that is the difference, which is the next question that we will answer, the difference in blood entering at A and leaving at B. So at A is oxygenated blood and more concentration of carbon dioxide, while at B is oxygenated and high concentration of oxygen. So that's the difference. List two structural number 3.15. List any three structural adaptations of the alveolus which makes it well suited for gas exchange. Number 3.15. Why is it more suited for what? For gaseous exchange. This we can also say that the wall of the alveolus is made up of what? Of a single layer. The wall of the alveolus is made up of a single layer. It is made up of a single layer. A single layer of squamous epithelial cells. Which making it very thin, making it very thin to promote the rapid diffusion of gas, making it very thin to promote rapid diffusion of gas. Rapid diffusion of gas, that is how it is adapted to perform its gaseous exchange. It is actually it is made up of a single layer of squamous epithelium cells, making it very thin to promote rapid diffusion of gas. That is diffusion of what? Of gases. That is the situation there. And then the alveolus is lined with a thin film of moisture. We are saying 2 is lined. It is lined with a thin film of moisture. It is lined with thin film of moisture. It is lined with a thin film of moisture, allowing for oxygen to dissolve. Allowing for oxygen to dissolve before diffusing what? Before diffusing. To dissolve before diffusing. So what we are saying is that it has got a thin film of moisture which allows oxygen to dissolve before diffusing, which is a quite good advantage also in terms of what? Of a gaseous exchange. And then the last one is that the alveolus is also cup-shaped. That increases the surface area for gaseous exchange. The last one is what? It is cup-shaped. It is actually cup-shaped, which we are so saying that it increases the surface area for gaseous exchange. to increase surface area for gaseous exchange. Let's just go to the last question. Inhalation is the process by which air rich in oxygen is drawn into the lungs as a result of the action of the breathing muscles, and then discuss how the process of inhalation occurs. How the process of inhalation occurs. How the process of inhalation occurs, which is question 3 point what? 3.2. 3.2. Question 3.2. Discuss how the process of inhalation occurs. How the process of inhalation occurs. It's quite a lot of points that we need to take note of here. The diaphragm contracts and flattens. 1. The diaphragm contracts. The diaphragm contracts. The diaphragm, it contracts and flattens. That increasing the volume of that would increasing the volume of the thorax from top to bottom. From top to bottom. And then external intercostal muscles contracting. External intercostal. The external intercostal muscles, they contract, causing the ribs to be lifted up. Causing the ribs to be lifted upwards. The ribs to be lifted upwards and outwards. And then the volume of the thorax cavity increases sideways. It increases. The volume of the thorax increases sideways. That is one other thing that we must take note of. So we are saying, actually, that is what to say that the external intercostal muscles, they contract, causing the ribs to be lifted upwards and outwards. Volume of the thorax increases sideways. And then one other thing is that the total volume of the thorax cavity increases and pressure total volume of the thorax the total volume of the thorax increases, and pressure on the lungs pressure on the lungs the pressure on the lungs decreases anyway. The pressure on the lungs decreases. Therefore, because since atmospheric pressure is now higher than pressure within the lungs, is now higher than pressure in the lungs. We are saying air rushes into the lungs, hence air rushes into the lungs. Air rushes into the lungs via the nostrils or air is drawn into the air passage through the lungs. So what happens is just a variation in the atmospheric in the volume of air, to say that the atmospheric pressure is now higher than the pressure in the lungs. Therefore, air moves from the high pressure into the low pressure, which is into the lungs via the nostrils. Therefore, it can be through the nostrils or the air passages into the lungs. That's how it occurs. That's how this occurs. So that's how the process of inhalation takes place. So you are just here asked to explain, to give a narrative of what happens during the process of inhalation. Differentiate the terms. Immigration and emigration. Let's just quickly rush to complete this one. Differentiate the terms immigration and emigration. Immigration is the movement of organisms into an area, while emigration is the movement of organisms out of an area. So in other words, we are just saying immigration is the movement of organisms. It is the movement of organisms into an area, while emigration it is the movement of organisms out of an area. That's what we call emigration. So immigration is movement into, and emigration is movement out of an area. And then the next question is what? The next question is natality and mortality. Natality is it is the inherent ability of population to increase. Is the inherent ability It is the inherent ability of a population it is the inherent ability of a population to increase as a result of new offsprings. As a result of new offsprings being added, which means being born, well, mortality refers to death of individuals. Death of individuals. It refers to death of individuals in a population. In a population species. So in other words, is natality is actually birth rate. An increase in population due to death, birth rate is natality, and then mortality, it is actually death rate of the death of individuals in a population species. That's what we call mortality. We have got a graph that represents population of zebras in a particular habitat. There we are given A, B, C, D, E, F, and population is given on the horizontal axis there, and then we have got time there. The question is saying, first question is saying, our first question wants us to say what is what? Um, okay, name the growth form shown in the graph. Name the growth form shown in the graph 3.3.21. Name the growth form that is shown in the graph. So the form of this growth, which is shown in this graph of this graph, it is called logistic growth form. It is called logistic growth. It's logistic growth form. It's logistic growth form in this case. We are saying it is logistic growth form. Identify phases A, B, and C. Identify phases A, B, and C. What is phase A, B, and C? So A, it is establishment phase. A, it is called establishment. It is establishment phase. Others, they also call it the lag phase. And then B, it is the geometric. It's the geometric phase. It is the geometric phase. And then C is also known as what? As equilibrium phase. C is equilibrium or it is called stationary equilibrium or stationary equilibrium or stationary phase. The next question is saying, explain the initial phase A. Explain the initial phase A. Start slowly. 3 point is 3.3.2.3. So in this case, what we need to understand is that population is adapting to the new environment. There are few sexually mature individuals. So population is adapting to the new environment. Is adapting to the new environment. And there are few sexually mature individuals. There are few sexually mature individuals. And then that is 3.2.2, and then 3.2, 3.3.2.4. What does the dotted line represent? The dotted line actually, it actually shows the carrying capacity. The dotted line, this dotted line, it shows the carrying capacity. The maximum population the habitat can sustain. This is the carrying capacity. Carrying capacity is shown by the dotted line. It is showing the carrying capacity class. It actually shows the carrying capacity. And then list 3 density dependent factors which should limit the size of the zebra population. 3.3.2. The next question, 3.2.5. What are those factors that can limit the 3 density factors which could limit the size of the zebra population? It can be 1, diseases. They can die as a result of diseases. 2, it can also because of predation. The zebra population can be reduced because of predation. Lions and cheetahs or whatsoever can eat them. And then we can as well agree to say that competition, it can also because of competition for food or what? Competition for food or space. Competition for food or space or mating partners. They can as well be factors that can also reduce the population of these. They can also be factors that can limit the population of these zebras here. Those are some of the factors. The next question, it's a diagram investigation to determine the size of crab population in a dam. 45 crabs 45 crabs were caught. The shell of each crab was marked with a spot of black paint. The scrap was then released back into the dam. 2 weeks later, a sample of 36 crabs were taken from the same dam. The following diagram, the following shows the crabs that were caught. The diagram shows the ones that were caught. You can see the ones which were caught and marked and sent back into the dam. So the question is saying, using the Peterson index, estimate the population, estimate the total population, the total number of the crabs in the dam. Show all your working using the that formula. So what do we say? How do we calculate this population here? How do we calculate the population of these crabs? We can therefore say that this is equals to we need to understand that this is equals to the population of these crabs or total number of crabs in second court. Let me just put it down there so then we can see it nicely. Or let me just put it here on top. We can say size of crabs. Size of crabs is equals to total number of crabs in second capture is equals to total number of crabs is equal to total number of crabs. Total number of crabs in second capture multiply number of crabs marked in first capture multiply by number of crabs multiply by number of crabs in first capture which we are saying over the number of marked over the number of marked recaptured in second capture, number of marked crabs recaptured in second capture. Recaptured in second capture. So in this case, we can just give to say that we have got our total number of crabs. We are saying total number of crabs in second capture, which is equals to, this is equals to 36. According to our context there, we are saying it's equals to 36. Sample is 36 multiplied by, we are saying multiplied by number of crabs in first capture, multiplied by 36 multiply by what? By 45 over, we are saying over the number of crabs captured, over the number of crabs recaptured in second capture, multiplied by number which is second recapture, which is close to 9. Over 9, which we are saying we get the answer is equals to what? 180. is equals to 180. That's how we calculate this one. The Mark-recapture method. I'm sure if you can refer to it, that you remember it very well, which can be summarized as to say, which can be summarized as what? As we can summarize this. Let me just use a different ink, which is equals to n. I'm saying it is n. N is equals to c times m over r, which is equals to 36 times 45 over 9, and then we get our what? Our 180 grams. So please take note of this, take note of this, take note of this. These formulas, I'm sure that you remember from the refer to the notes also. There is a question that I did on predation and stuff. Can we also refer to our notes on the channel also? Refer to the notes on the channel also. I'm sure there is a question which talks about the mark and recapture method. N is equal to C times M over R, which we get 180 there. The next question is saying, suggest 2 reasons why the estimated size of the population may differ from the real population. Why the estimated population may differ from the what? From the actual population. One, it is because question 2.1, we are saying it is because some of the crabs may have left the area. Some of the crabs may have left the area, meaning to say that they may have migrated. When they saw people entering there, they may have moved out of the area. That can cause them to have a different population. And then some crabs may have left the area, or some may have died. Some may have died there. And then we are saying also estimate is unreliable because only one sample is taken in this regard. Estimation may be unreliable, It can be unreliable because only one sample was taken. Unreliable because only one sample was taken. So that is how we can make also this population not to be correct. Last question is saying list. The last part is saying list 3 precautions to be taken to ensure that the mark recapture method is reliable. The mark must not harm or affect the animal's behavior, movement, or acceptance within the population. 1, marks must not interfere with the organism's lifestyle. It must not interfere with the organism's lifestyle. The marks must not interfere with the organism's lifestyle. And also, which is to say the organism must not die, it must not be affected in terms of its breathing, its movement, and acceptance by other organisms in the what in that place. And also we can as well say that the mark must last for entire period of what of investigation. Marks must last for the entire period. They must last for entire period of the investigation. And then we can as well say that sufficient time must be. We can also say that sufficient time must be allowed for the marked animals to mix with the population. Sufficient time must be allowed for the marked to mix with others. So we can say these are some of the reasons that we can give there in terms of our marks that we can give in regard to this, in order to ensure that the mark recapture method is reliable. Those are some of the things. And it must also take into account also that it must also take into account factors such as that the chances of the animal being caught must not be affected by the method of the capture. Also, the method of capturing must not affect the animal that is being caught. That is one other point that we can give in this respect. And then the population must be closed. The population must be a closed population. That is, no immigration or emigration must take place during the sampling, and the count must not take place during the breeding season. It mustn't take place during the breeding season, and also the area or the habitat in which these organisms are kept, it must be controlled to avoid immigration and emigration. So therefore, the population is confined or is barricaded. I'm sure that you would get this so useful. Thank you so much for your time, and please don't forget to share, to subscribe, and also to comment and engage into this posting, so then we can do more than this. Keep connected for the next upcoming videos. Thank you so much for your time, and have a beautiful day.